Yes, good morning. I apologize, I speak in English. So, right when they invited me, I had 2 months, but it was too short to learn Spanish. So, I hope it's not a problem. I will try to speak a bit slowly. So, thank you for this great introduction. I really enjoyed being here, and I would like to thank the organizers, Monica, for this invitation, which gives me the opportunity to come again. And I say again, because I was here exactly 10 years ago, and when Professor Rogelio invited me to give lectures here for the students, so I had a 3-day course with these great students. Maybe there are some, recognize yourself here. That was a really great time. I really enjoyed, because not only because it was nice, but the enthusiasm of the students, that was really something special. So, and that's why when I heard that I have to come again, I did not have to think long, so I said immediately, yes, I'm coming. So here I am. So just to give you an idea where I'm from, yes, so this is the city where I'm from. This is Delft, a very small city, just between Rotterdam and Den Haag. It's a beautiful city, which used to be the biggest city in the Netherlands, but now it has only 100,000 inhabitants. So compared to Mexico City, this is just a beautiful village. It does have okay. It does have a great names coming from this city. So Johannes Vermeer, you know this painter, maybe. But Anthony van Leeuwenhoek, the inventor of the first microscope, we are very proud of him, because he used to live in Delft many years ago, of course. Both are buried in Delft. So Delft is also famous for this blue work. Delft has also a very big university, so there are 8 departments, and I am here at Applied Sciences, between departments Biotechnology and Radiation Science and Technology. These 2 buildings just next to each other, and this is in these 2 buildings because my research concerns activities going in these 2 departments. So there I have my group that works on agents for imaging and therapy, but I'm a chemist by training, so we approach this from a chemical point of view. So we design agents that are able to visualize a disease, mainly cancer, and also we hope to have a system that will treat at the same time. I brought a few examples here for you to just to demonstrate what we do, and to get your interest to this field, maybe. So first, of course, when we design agents to do imaging, it is important to understand what kind of imaging we are talking about. Because if you go to the hospital, sometimes doctors need to send you to a scanner, right? And depending on what they need to visualize, they will choose a certain technique. And every technique has a particular ability to visualize some events in the body. So here you can see, for example, computed tomography or MRI, molecular magnetic resonance imaging, and ultrasound. They have perfect anatomic images, so they have high resolution. But if the doctor wants to visualize some inflammation or some processes that go on a cellular or even molecular level, then these techniques are not suitable. So for these, you should go to positron or single photon emission tomographies, which are very sensitive, and the optical imaging is also very sensitive on molecular level. But of course, to visualize a human by optical imaging is not realistic, because the optical signal has a penetration depth. Right, so it is when we design contrast agents, of imaging agents, it is important to understand the signal of origin of the signal. So, for example, in in nuclear imaging or optical imaging, it is clear that we need the tracers or optical probes to get the signal. In case of MRI, which is a very high resolution technique, so very valuable technique, we are not looking at the agent itself, we are looking at behavior of water in our body that behaves in different ways depending if it's a healthy tissue or a diseased tissue. And for that we use contrast agents. So what contrast agents do, they influence behavior of water molecules in our body. And so this is where our works come in, but just for you to understand better, I don't know if you're familiar with nuclear magnetic resonance, NMR technique, exactly the same principle. So we have a patient here, which is placed in a magnetic field here, and so the protons in our body align with magnetic direction of magnetic field. And then we give a radio frequency pulse, which flips our protons in one direction, and when the pulse is taken away, then they relax back. And that is what's happening in our body in MRI scanning. And what happens when we use contrast agents, so this process of relaxation, either T1 or T2 relaxation, can be influenced by the presence of contrast agent. And in this case, we can visualize things that were not visual before introduction of contrast agent. Right? So for this, these are three typical molecules that are used in the clinic. So patients are injected with this. And here you have metal ion in the middle, which is paramagnetic gadolinium, complexed in a small molecule because it's toxic. And there is one water molecule which is exchanging all the time. So when we as chemists look at how to improve these contrast agents, then we look at this schematic representation. So here is the chelate, the gadolinium in the middle, and then now it goes by itself. And then this water is exchanging, and these parameters, they give you the efficiency of a contrast agent. So what can we do as chemists? We can make sure that there are more water molecules coordinated to one ligand, one complex, that will increase the signal, of course. Or we can make this water exchange faster, that will also work. Or we connect this small molecule, small complex, to a large protein, large polymer, which means that the molecule will move slowly, and it means that the relaxation, the signal will be strong. But all these modifications, 20 years of research showed that we cannot do that. We cannot improve too much because whatever we modify here in the molecule compromises stability of the compound, and the gadolinium starts migrating away, and that it becomes toxic. So what we do instead, we use different approach. So we use nanoparticles. Let's see. Nanoparticles that in this way we can deliver many gadolinium ions to the site of interest and improve the signal. Or we can use targeting vectors, which means that have special molecules that find tumor in the body and bring contrast agent there. Or we can combine with more sensitive techniques. So there are a couple of things that we can do. And I wanted to show you a few examples in this direction. Okay, so for this, in my group, I use lanthanides. So these are very interesting elements. They are all have the same chemical properties, all 14 elements, but they all have different physical properties, which means that we can plug and play, we can just combine them, and then we can make multimodal nanomaterials, right, when we exchange these elements. So that gives, of course, some thinking work for a chemist, and then thinking of this, we come up with the nanomaterials, because these are the most versatile materials to incorporate different functionalities. So just let's refresh what is nanotechnology. First, what is nano? Nano means, in Greek, means dwarf, which is very small, so a billionth part of a meter. And then the comparison that I make, if nanoparticles would be as the size of a ball, then a mice would be the size of Mexico. So that is just to have this in our imagination. Concerning technology, what is technology in this case? So is it chemistry, is that physics, engineering, biology, all together, and then on a nanoscale, on a scale of from 1 to 1,000 nanometers gives us nanotechnology, and that combines all the sciences resulted in a product. And if we look at around us, then we find nanotechnology everywhere. In all this, I will not read them all, because we are interested in this, in medicine and drugs. And in particular, with my talk today, I focus on cancer. So what can nanotechnology do for cancer, or better to say, against cancer? And, okay, for these first, some scary things. We know that it's a horrible disease, it takes many lives, and if we don't come up with a brilliant approach, then these numbers are only growing, and in 2040 the predictions are. And if we focus on particular cancer, which in my projects I work a lot with breast cancer, so the numbers are also dramatic, and just to be gender equal, you should realize that men also can get breast cancer, although of course much less than women. But why I'm saying this, to look at this scheme, that depending on which stage is the tumor, the different treatment is needed, of course, and that again from a chemical point of view, it involves different type of chemistry, different approach. And so I put my talk in such a way to show you that, okay, so what can nanotechnology do for these different stages in oncology? Of course, when the tumor is compact and is located somewhere that is detected and is not spread, it's very easy just to cut this thing out and get rid of it and then done, right? That's the best way. And for this, okay, what can nanotechnology mean for this? So you have such, I don't know if you see the images well. So here you have an open place, the surgery on a tumor, which the surgeon has a very vague idea where the tumor is, just to be sure it cuts too much, just to be sure that everything is cut away, which in case of brain tumor can be very dangerous, of course. But here, in the new surgeon view, after introduction of this large molecule, this is highlighted, and then the surgeon performing under microscope can cut away this particular part. Okay, so what happens when the cancer is in more advanced stage? So then different approach is necessary, and this is the delivery of therapeutics. It can be hormone therapy, can be chemotherapy, and of course the idea is to deliver it locally in order not to harm the whole body. And so for this there are several strategies. One of those is passive drug delivery, which means that tumor is growing very fast. So the blood vessels around tumor don't have time to mature, so they're growing very fast, and it means that the walls in the blood vessels have holes. So when nanoparticles are passing through the blood vessels, they extravasate and they remain in the tumor. So this is a passive way to deliver, so we need vesicles to bring the therapeutics to the site of interest, and for that, in many cases, liposomes are used. So these are double-layered balls that contain drugs in a core. So here we have some results. We designed long-circulating liposomes, and we designed them in such a way that they would release their content only in the place which was heated up, and heated up to a very sharp temperature window. So what we used for that is we designed a special way to build the bilayer using these two lipids, and were supported by cholesterol to make them strong. And when we play with the ratio, we can design in such a way that they become labile at certain temperature. So we used then these two molecules to label the rhodamine to label the membrane so that they're always visible in the red color. And then we put calcium inside, which is silent inside because it has a high concentration inside and there is self-quenching, but once it is released, then you get the bright green signal. And so we did this intravitreal microscopy. So you see here a mouse with a skin, in a back skin. There is a window placed and there is a tumor between these window glasses. And then we injected these liposomes and so they are circulating. You see red color. And then they passed this window which was heated and there is a release of the calcium which becomes green because it is released and concentration outside of the vesicle is lower. Okay, so this is the way how deliver the drug. So this is used also in clinics in a very nice way. Here. So, and what if we need to deliver drugs to the target and we want to use special vectors, special overexpressed receptors that are present on cancer cells, and we want to send our agents directly there. So this work is very special for our group. What we look at the cell membrane in the terms of on a molecular level, and we know that there is one sugar at the end of the glycolipids and glycoproteins which cover tumor cells, and we can target that. And we can target that, this is called sialic acid, and we can target that by this chemical covalent reversible chemical binding. So, okay, this is something I'm going to run in the slide. But we designed such an agent here. This is just a normal MRI agent which is modified. Okay, this I don't know what it is. Should not be with this. So it binds. Go back. It binds this part of the sugar very selectively. So it took a while until I found a place where I could do in vivo experiments for that, because the regulations are very tough in the Netherlands. So with the partners in Turin, we did this work, and then we showed that when we injected this designed molecule, we could target the tumor. So you see here is the tumor with a normal contrast agent, it's not visible. And here is the tumor after injection of our designed agent, and it goes immediately to the tumor. And it remains there for 24 hours and then it goes away. And this is the comparison with just a normal contrast agent. So you see how different the accumulation is. So that was a very good result, of course. We also labeled this compound with a radio label, and then we could observe with different imaging techniques, which is more sensitive, and then we could observe the same for different type of tumor. Okay, so that work was very promising, and that was where our collaboration started with Dr. Refilio, when he came to the Netherlands for 1 year, and he did wonderful work as a synthetic chemist. So he expanded this molecule, making it better. So what we did there is introduce the 2 fluorine atoms in the molecule, which means that it brings the pKa of this group more suitable for physiological pH. You see here, which means that more binding would be possible with this new ligand. And we also developed new method as a solid phase synthesis, because the boronic compounds are very difficult to synthesize, or better to say, very difficult to purify after the synthesis is done. So in this way, the synthesis was happening on the resin, and then only at the last point, when the product was there, then it was cleaved. And that this principle is used for peptide synthesis. We applied that for these small molecules, and it worked. And we went further. We prepared this kind of molecules when there are 2 gadolinium, which of course means better signal. So you see here this is a higher signal compared to when there is only 1 metal ion in a complex. Okay, so the safe delivery of drugs is of course very important. And that becomes even more important when we talk about radiopharmaceuticals, because radioactive compounds, you don't want them to circulate too long in the body, you want just to shoot to the target, right? So the faster we can do it, the better. So first, just for your general knowledge, what is radiation therapy? So it's basically the strategy is to damage DNA, and the more DNA breaks we can introduce, the more effective it is. And it can be direct, so the DNA can be killed, destroyed directly, or indirect, that you generate the oxygen reactive species, which then damage DNA. But in any case, the way that is done in clinics at the moment is this. So the patient is there, lying under the equipment that produces radiation, X-ray radiation that is directed to the tumor, and nowadays this equipment is very specialized. Of course, it hits the tumor very precisely, but nevertheless, it doesn't exclude the fact that the X-ray should penetrate the skin, the derma, all kind of tissues before it reaches the tumor. And in case of breast cancer, the danger is to hit the heart, of course, and in case of brain tumor, again, very vital, millimeters can make a difference, right? But this is not our piece of cake. As a chemist, we want to design agents that would be suitable for internal radiotherapy, which is at the moment is not used in clinic, only for terminal patients, just to decrease their pain, but not really to cure, with some little exceptions. I will show you those. Okay, so we want to design an agent which is containing radioactive ion that is going to the tumor directly. And I show you this, again, another lanthanide, so talking about plug-and play. So here it is, holmium, which is very interesting because it has three functionalities. It has very strong magnetic moments, so it can be used as an MRI contrast agent, but it also has X-ray properties when it is a nanoparticle, and it can be activated by neutrons, which means that you can go from one isotope to another isotope without changing the chemistry, and it's an easy process. And it is suitable for radiotherapy and also for imaging, radionuclide imaging. So great element. An example from clinic, which is already in the Dutch hospitals, even paid by insurance, which means that is good. Designed by our colleagues. So holmium microspheres, they are injected in hepatic artery, and just for you to know, hepatic artery, this is the blood vessel that brings blood to the liver, period. Nowhere else, just to the liver. So if you inject in this hepatic artery, they jumped, they were supposed to. Okay, so if you inject these nanoparticles, then because they're big, they're not nano, they're microspheres, so, and at a certain point they stop, because the capillaries get smaller, so they cannot pass anymore, and radioactive microspheres, they remain there, and they kill the tumor. Beautiful methods for liver tumors, but of course limited to that. And microspheres are also very difficult to handle because they stick to the needle and disaster. So what we propose is to make nanoparticles. Nanoparticles can circulate, they can extravasate the leaky blood vasculature, they have a very high MRI signal, and we have all the methods to produce nanoparticles of lanthanides of different sizes, depending on the method that we apply. And if we decorate properly at the surface, we can make sure that they go directly to the tumor. Of course, it's a beautiful design. What about the logistics? So here I'm missing colors again, but okay. So what we have here is our targeting vector, this phenylboronic acid. The polyethylene glycol, I will show you later why it's needed. The fluorescent label, and then some polyarginines to make sure that this penetrates the cells. And So, and what we did, we activated these materials. Why I show this? This is important because normally you would use radioactive polymer to prepare nanoparticles, and then this is the process of using radioactive material during reaction. This is very, not very convenient. So what we did, we put everything on a nanoparticle and then we activated, and it worked. We saw that we don't lose fluorescence, which means that this molecule is not damaged, which means that the others are also not damaged, and we saw here that they do accumulate with the cells. So here around the nucleus, which means that the penetration mechanism worked as well. So, and that means that for the clinics it will be very advantageous because we can produce these nanoparticles very easily. Now I saw that I have 10 minutes, so I don't know if I will be able to show you this part, but maybe just short. Because we do work with porous materials as well, and they are very interesting, very promising, because you can do them used in medicine for many good applications, like releasing these drug molecules from the pores. But we work on these microporous materials, which are beautiful ones. And yesterday, Dulce explained to you that these are also polymers, this zeolite material. So now I dare to show it to you. So this particular one is LTL nanozeolite, which has this beautiful framework that you can load with many lanthanide ions, gadoliniums in this case, and then they can do the work. So here we use the first europium, which is an optical element. So we load this europium in the big cages, then we calcine this material, which means heating up to 600 degrees. And what happens then, that this europium migrates to small cages and stays there irreversibly. And while this is happening, it loses its water molecules that are coordinated around, and it becomes fluorescent, luminescent. So then we add gadolinium to the big pores, and then because it has 6 water molecules, like I said in the beginning, that the more water molecules, the higher the signal, then we get here a very high MRI signal. Okay, so so this is for you to see how big is the signal increase due to this strategy. And of course we wanted to put these materials further in research, and what we needed to do, I will show you this, the need of pegylate nanoparticles, which means to put polyethylene glycol on the surface of nanoparticles. And this work was done for the first time many years ago by this gentleman who passed away last year in the age of 100. But you don't see it, but here I found it fascinating. He wrote here, in the 60s I was between the grants, nothing to do basically, I had no money to do any research. And I became interested in these molecules, and he discovered that if you put these polymers on the surface of nanoparticles, then the immune system does not recognize them, and that it allows these nanoparticles to circulate longer and to find the tumor. It is a very important piece of research that allows us to do this. So we provided our nanoparticles with this long polyethylene glycol chain to increase their circulation. And I'm just skipping because of the time. I just show you, we labeled it with radioactive zirconium to be able to visualize it in a more sensitive way. And we injected these nanoparticles in the mouse. No tumor, just to see what is happening there, and we found them all in the lungs, and then in a time they were passing out, they were passing to the liver. So we did biodistribution, and we discovered that they all stayed together because the composition in the end was the same as in the beginning, so nothing was leaking, and this is a very promising probe to do imaging and therapy of cancer. So here we provided them with the folic acid, because tumors also overexpress folic acid receptors, and we labeled them also with the radioactive copper, which is therapeutic radionuclide, and we saw that we can destroy tumor cells having this system on one nanoparticle. So, in conclusion, what I showed you has been very, very fast and very. I apologize for that. I wanted to show you more. So I showed you that we can go from fundamental understanding of the materials to real products. Of course, we do need clinicians for that to guide us, to tell us what they need so that we can make it. Because clinicians are very stubborn people. They think that they know everything. They don't need new materials. You come to them with a proposition and they say, oh, we already have this. We don't need a better one. So our goal as a chemist is to show that we can improve, we can design beautiful things, especially using nanosystems like polymers, and they play a very important role in this path. So with this, I want to acknowledge all these people that participated in this work and all this funding that made it possible, and I want to thank you for your patience, for me, for your attention, and also for your interest, and I wish you all the best in your careers, in your studies, so that you get inspired. I hope I contributed a little bit to this process, and I will take questions. Thank you.
Vamos a agradecerle con un muy fuerte aplauso, a la doctora. We will proceed with the questions. So, if you like to answer, if you like to question about in English, or if you like, we can translate you. Nosotros podemos traducir si ustedes quieren preguntar, no todo, pero vamos a hacer nuestro mejor esfuerzo para que también la doctora, no sé si la doctora también pueda entender un poco de español, si usted habla lento, ¿lo entiende?
Thank you. Good morning. You mentioned liver cancer, but I have a question. Is this limited to one or some types of cancer, or can this be applied to a much larger variety of cancer types?
This, what I showed you, is exactly concerns only liver cancer, because there is no other example that the organ has its personal blood vessel. There is no such a case, and that's why, because the strategy is to inject in the blood vessel that goes to the tumor, and that is only in case of liver. So that's why it's limited to liver cancer. Thank you.
Bueno, aprovecho, Dina, la oferta de hablar bueno, ya que ustedes nos traduzcan, así que me gustaría saber, bueno, menciona algo de que los tintes químicos que se usan para las células, para teñirlas, que pueden llegar a ser un poquito tóxicas. No sé cómo se garantice que, pues bueno, que no sea realmente dañino.
But which ones? I mean, every you mean imaging agents, right? So every imaging agent has toxicity. In fact, these MRI contrast agents, which were used already for, I don't know, so many years, only now was discovered that gadolinium, we knew that these agents are dangerous for people with kidney problems. We knew that. So if you would go to the hospital, you would need an MRI scan and you would have just a component kidney problem, you would not be injected with this agent. So that we knew, but the recent discoveries show that also there is some accumulation of gadolinium in the brain. So now the whole research society turns away from gadolinium, going to manganese. Manganese is a better element because we have it. This is an endogenous element. And it is also paramagnetic. not that effective as gadolinium. So what I mean is that checking the toxicity is very important, and many beautiful designs that we come up with as a chemist end up in the garbage because it's too toxic. That's a good point.
Entonces, que sí podemos, lo que no sea reconocido por el cuerpo, pero es que los investigadores están viendo de gadolinium a magnesio, que es un elemento más amigable con el cuerpo humano. Another question?
Hello, good afternoon, thank you for your conference, and I would like to question. Currently, the medicine don't have not the same effect like before, and these nanoparticles can increase this effect in the medicine.
You mean for therapy or for imaging?
In general.
They can, for imaging, they can increase the effect because one gadolinium can do, 1,000 gadolinium can do more, right? So this is what I meant by high payload of material, which nanoparticles do. They bring a lot. Or the zeolite porous material, we can load with many ions, and then they can amplify the effect, of course. But another problem is that the toxicity of nanoparticles has to be considered very carefully, because you can do the therapy, but what happens after a couple of years, right? When the patient is maybe retaining these nanoparticles, how harmful would this be? So these are, you can treat better, but you can harm later, maybe. So we can think of all of this in the design. Yeah.
Thanks.
Good morning. We have in this moment we have different therapy of nanotechnology, exists cisplatin, docetaxel, nanotechnology for the cancer. But the big problem in this is the scalability of the nanotechnology for the manufacturing product. Do you think that this investigation have the possibility of the good manufacturing for traduce this investigation in manufacturing product?
This is a very good question. Yeah, this is something that we tend to forget, because when we do something nice, which gives nice signal or nice image, we don't think, okay, so then put it to the patients. Then how do you produce it? Just to give you an example, a very recent project that I mentioned that we work on breast cancer was granted exactly to develop new breast cancer therapy by nanoparticles, and there was even a company involved in this process, which was very good, of course. And then the pandemics happened, and then the war with Russia happened, which meant that we wanted to use a radioactive palladium, and it means that 99% of the world's radioactive palladium was coming from Russia. So the whole project, we published, we worked on that, we developed things, but then it is useless, because we cannot put it in a production, because it has it's not possible. But okay, this is an extreme example. Another example is what you are asking, that easier nanoparticle. How sure can you be that it can be manufactured well? And this is a difficult process, because nanoparticles in general, the disadvantage is that it's very difficult to control exactly the size, it is nanoparticle of this metal for sure, and you know approximately the size, but once you inject, it starts behaving differently, because you always have a distribution. So this manufacturing process is very important, is difficult, and unfortunately in my career I never came to the point of talking with manufacturers and solving this problem, so I'm aware of this, but before you reach this point that industry wants to produce, you need to show so many things in preclinics, clinics, phase 1, phase 2. So that's why this large part of research ends up in the clinic only a very small percentage. And still we are trying.
So you think that the stabilization
Stabilization in which way? Colloidal stability or biological stability? Different
the
things.
stabilization of the, or make the drug release.
Colloidal stability is also a difficult point, yeah. Yeah, because you will need, it is very difficult to keep them stable on a shelf. And then so you need trained personnel that would make the suspension. That's another problem. Yeah. Thank you for the question.
Bueno, gracias. Todavía tenemos tiempo para otras preguntas. Si alguien quiere preguntar.
Thank you very much. Sorry about my English. I hope that don't break your ears. Well, I have a couple of questions. The first is I didn't understand why are you using polyethylene glycol? It's about the hydrophilic. Yes, it's about that.
Is one to help to stabilize, and another thing is that when the nano objects, like foreign objects, are injected in the body, you can compare it with the allergies. Why people have allergies? You breathe something that does not belong, and then the body reacts, sends macrophages to remove it and to put somewhere to disarm. The same mechanism. So there is something foreign floating in the blood. The immune system goes there, takes it, and puts it in the liver. Liver is our filter, right? So when you have polyethylene glycol, then it takes longer until the immune system has the signal. I have to work.
Okay. Thank you very much. And what about the, sorry, I don't know a lot about nanoparticles, but I have the idea that when you inject, the nanoparticles can suffer an effect and destroy or even agglomerate. Is this correct?
Of course they can. Polyethylene glycol helps against this, so they don't agglomerate. So this is one part.
Yeah, okay. And about the markers, because you use it like imagining, yeah, remember, or like a drug delivery, and you say that you want or you modify the particles to target exactly. But how can you? It's only chemical. It's only the chemical effect that your nanoparticle is going to target, or you have another effect,
Two effects. One effect that nanoparticle has, that it goes through the blood vessels because the blood vessels have holes. This is passive delivery, and then they stay there because of this chemical binding. So there are two effects together.
Very interesting. Thank you very much.
Thank you.
Wonderful.